When construction workers broke ground on a small Caribbean island in 2010, they were not expecting to find anything unusual. They were digging in an ordinary commercial district in the capital city of Philipsburg on the island of Saint Martin, but a few feet below the surface, their equipment hit something that would stop everything and eventually reach the laboratories of Stanford University and the University of Copenhagen. 3 sets of human bones, very old bones, no names, no records, no grave markers, nothing to tell the world who these people were, where they came from, or how they ended up beneath the streets of a Caribbean city. But the bones had something that written records could never erase, something invisible to the naked eye, sealed inside the hardest part of the human body, the teeth, something that had survived 3.5 centuries underground, through heat, humidity, and the slow decay of everything else that had once made these people human, their DNA. And when scientists extracted it, sequenced it, and compared it against the genetic profiles of hundreds of living populations across the African continent, what they found did not just identify 3 nameless people. It cracked open a door that scientists had been trying to open for decades, and revealed a truth about the slave trade that no ship manifests, no colonial record, and no historical document had ever been able to tell. Before we dive in, comment where you are watching from and click subscribe. To understand why this discovery was so extraordinary, you first need to understand the problem that had haunted historians, geneticists, and the descendants of enslaved people for generations. More than 12.5 million Africans were forcibly taken from their homelands and transported across the Atlantic between 1515 and the mid-19th century. Some 2 million of them died during the crossing, during the brutal ordeal known as the Middle Passage. Those who survived arrived in the Americas stripped of almost everything, their names in many cases changed or discarded, their languages suppressed, their family connections severed, their ethnic identities erased. The records that do exist, ship manifests, colonial documents, and port logs tell you where a ship departed from in Africa, but they almost never tell you where the specific individuals on board were actually born, which ethnic group they belonged to, or which community was shattered when they were taken. For the descendants of enslaved Africans living in the Americas today, this has meant something deeply painful, a wall in their family history where their ancestry simply stops. A place where the records run out and the names disappear, and the question of where they really came from goes unanswered. Unlike the descendants of European immigrants, who can often trace their lineage back to a specific village or country, many African Americans, Afro-Caribbeans, and Afro-Latinos hit that wall somewhere in the 1700s or the 1800s and find nothing. Not because the people were not there, but because someone deliberately built the wall. DNA science has been slowly, painstakingly chipping away at that wall. The bones found beneath the streets of Saint Martin were one of the first major breakthroughs. The research was led by a team that included geneticist Carlos Bustamante of Stanford University and ancient DNA specialist Hannes Schroeder of the University of Copenhagen, 2 of the most respected names in ancient genomics. Their task was immense. The bones had been buried since the late 1600s in a tropical environment. Heat and humidity are the enemies of ancient DNA. They break it down, contaminate it, turn it to biological noise. Many ancient DNA projects fail completely under such conditions. But the teeth held. The team used molecular techniques specifically designed to pull genuine human DNA from samples that have been invaded by the microbial genetic material that floods bones after death. A method that allows researchers to find the real signal in an overwhelming amount of biological noise. From the DNA they recovered, they were able to sequence enough of the genome to do something that had never been done before with samples this old from a tropical environment. They identified, with high specificity, the ethnic origins of the 3 individuals. The results told a story that no document had preserved. Carbon dating confirmed the 3 individuals, 2 men and 1 woman, had died between 1660 and 1688. That placed them at the very beginning of the transatlantic slave trade's most brutal expansion. But the DNA told them something more precise. Comparisons against the genetic profiles of living populations across 11 African ethnic groups showed that one of the individuals was most closely related to the Bamoun, a Bantu-speaking ethnic group from northern Cameroon. The other two showed strongest genetic affinity with populations from Nigeria and Ghana, including groups related to the Igbo and the Brong peoples. What the shipping records had recorded as the general departure of enslaved people from West African ports, the DNA translated into actual human beings with specific homelands, specific cultures, specific identities, people whose descendants are alive today. Bustamante, reflecting on the significance of the findings, said that through the barbarism of the Middle Passage, millions of people were forcibly removed from Africa, and what science could now do was begin to answer who they were, where they came from, and who today shares DNA with those people taken aboard the ships. But the St. Martin discovery, groundbreaking as it was only the beginning. Far out in the South Atlantic Ocean, 1,000 miles off the coast of Southwest Africa, sits a remote British territory called St. Helena. Most people know it as Napoleon's final place of exile. Far fewer people know it as the site of one of the largest forensic investigations into the identity of enslaved Africans ever attempted. Between 1840 and 1867, the British Royal Navy was patrolling the Atlantic, intercepting slave ships and freeing the captives aboard as part of Britain's effort to dismantle the transatlantic trade. Over nearly 3 decades, roughly 27,000 Africans were taken from seized slave ships and deposited on St. Helena. The British called them liberated Africans. But liberation, in this case, was a complicated word. Housed in ramshackle tents in a barren, arid valley called Rupert's Bay, the people arrived sick, malnourished, and traumatized from the sea crossing. Between 5,000 and 8,000 of them died there, on an island they had never heard of, far from any home they had ever known, and were buried in mass graves. For more than a century and a half, those graves were largely forgotten. Then, in 2007 and 2008, ahead of roadwork and the construction of St. Helena's first airport, archaeologists excavated the burial site at Rupert's Valley. What they found was heartbreaking in its scale: rows of burials, many with no individual markers, representing people who had survived the slave ships only to die before they could reach any life resembling their own. Some skeletons showed signs of dental modification, the deliberate filing or chipping of front teeth into distinctive shapes, a practice tied to specific African ethnic groups and cultural identity. Some individuals were buried with small objects, beads, necklaces, personal items that they had somehow kept through the ordeal of capture and sea crossing. Artifacts of identity, evidence that even in the worst conditions imaginable, people held on to who they were. This is the part of the story where a team at the University of Copenhagen, led by paleogenomicist Marcella Sandoval Velasco and ancient DNA researcher Hannes Schroeder, the same Schroeder from the St. Martin discovery, spent years attempting the nearly impossible. They tested remains from 63 of the individuals buried at Rupert's Valley, looking for intact, ancient DNA. Working with colleagues from Decode Genetics, the University of Iceland, and several other international centers, they managed to sequence partial genomes from 20 of the 63 individuals. The results, published in the Sunken Journal of Human Genetics in September 2023, provided the first direct genomic evidence of where St. Helena's liberated Africans actually came from. Their genomes were compared against genotype data for more than 3,000 present-day individuals from 90 populations across sub-Saharan Africa. The finding was that the individuals most likely originated from different source populations within the general area between northern Angola and Gabon in West Central Africa. The DNA clustered most closely with present-day Bantu-speaking populations from that region, populations whose descendants still live in Angola, Gabon, and the Democratic Republic of Congo today. There was one more finding hidden in the data, one that confirmed what historians had long suspected but had never been able to prove so cleanly with biological evidence. 17 of the 20 individuals whose genomes were successfully sequenced were male. Only 3 were female, This matched documented records showing that in the final decades of the transatlantic slave trade, men were being taken at a dramatically higher rate than women, a sex bias that the DNA preserved in cold molecular evidence, centuries after the records were written and the ships were long gone. If you are finding this as important as I do, this kind of forensic science giving names back to people history tried to erase matters, subscribe to this channel. Every video we make exists to bring this research out of academic journals and into the hands of people who deserve to know it. Now, you may have heard about the Clotilda. If you haven't, the story is almost too audacious to believe. In 1860, 50 years after the international slave trade had been made illegal in the United States, and more than 2 decades after Britain had outlawed it entirely, a wealthy Alabama businessman named Timothy Meagher made a bet. He wagered that he could smuggle enslaved Africans into the United States undetected. He hired a ship captain named William Foster, who sailed an 86-foot wooden schooner called the Clotilda to the port of Ouidah in present-day Benin, and returned with 110 captive Africans locked in the hold. Once the enslaved people were unloaded under the cover of darkness in the Mobile River, Foster set fire to the Clotilda and sank it. It was an attempt to destroy the evidence of a crime that was, by then, a federal offense carrying the death penalty. The ship stayed at the bottom of the Mobile River for 159 years. Then, in 2019, archaeologists found it. It was more intact than anyone had dared to hope. A state-funded research project was launched. Marine archaeologists, forensic scientists, and DNA specialists descended on the site. Among the most urgent questions was whether human DNA could be recovered from a wooden ship that had been underwater for over a century and a half. Could skin cells, fingernails, hair, or other biological material deposited in the hull during the terrifying crossing still hold genetic information? Frankie West, director of the Forensic Science Program at Western Carolina University and the lead genetic analyst on the project, funded by the National Geographic Society, explained that cellular material could potentially be part of the accumulated sediment on the bottom of the hull. DNA might still exist in the vestiges of biological material deposited by the captives during their entrapment. The research is ongoing, but what the Clotilda has already confirmed is profound in a different way. The 110 Africans who were taken aboard that ship, people whose names we now know because they survived, were freed by the Civil War and went on to found a community called Africatown, north of Mobile, Alabama, finally have their story grounded in physical, archaeological, scientific reality. Africatown still exists today. Descendants of the Clotilda's captives still live there, and they have spent years fighting to preserve it against industrial pollution, against neglect, and against a highway that cuts through the heart of the community their ancestors built. The recovery of the ship, combined with ongoing DNA work and genealogical research, is slowly restoring a level of documented identity to those 110 people that the slave trade tried to permanently destroy. But perhaps the most sweeping piece of DNA science to emerge from this entire field came not from a single shipwreck or burial site, but from the genetic profiles of more than 50,000 living people. In 2020, researchers at consumer genetics company 23andMe published a landmark study in the American Journal of Human Genetics. They compiled genetic data from consenting participants with African ancestry across North America, Central America, South America, and the Caribbean, and compared it against the historical records of the transatlantic slave trade. The scale of the study was unprecedented. The findings cut right to the bone of what the slave trade actually did to human beings. First, the expected findings, the ones that confirmed history. Nigerian ancestry turned out to be significantly overrepresented in the United States compared to what shipping records predicted. The researchers consulted historians, who provided the explanation. Enslaved Nigerians were frequently transported first to the British Caribbean before being sold again into the American South, a slave trade within the Americas that the genetic record had quietly preserved long after the paper records fell silent. Ancestry connected to Senegal, And the Gambia, on the other hand, was notably underrepresented, with fewer genetic traces than ship manifests would suggest. The likely explanation is devastating. Enslaved Senegambians were disproportionately sent to rice plantations in the American South because they had expertise cultivating rice in their home regions. Those plantations were rampant with malaria. The death rates were extreme. The DNA evidence, centuries later, carries the biological signature of that mortality. But the finding that most shook researchers, and that no one could ignore, was this: African women had contributed far more to the modern gene pool of the Americas than African men, despite the fact that men made up more than 60% of all enslaved individuals transported across the Atlantic. In North America, African women contributed between 1.5 and 2 times as much genetic material to the modern population as African men. In Latin America, the numbers became staggering. In Central America, in the Latin Caribbean, and in parts of South America, African women contributed between 13 and 17 times more DNA to the modern gene pool than African men. The researchers were explicit about what this meant. The sex bias in gene pool contributions was attributed in the study to the rape of enslaved African women by slave owners, other forms of sexual exploitation, and deliberate policies in some countries, particularly in Brazil and several Latin American nations, that encouraged the reproduction of enslaved women as a strategy to maintain the workforce as abolition approached. The DNA was not just telling you where people came from, it was documenting, in molecular terms, the systematic rape of millions of women. It was recording crimes that were never prosecuted, never acknowledged, and in the language of biology, never erased. This is what makes slave-ship DNA science different from most other fields of genetic research. Most ancient DNA work reconstructs population migrations. It maps the movement of the peoples across continents over thousands of years. It answers questions that are scientifically interesting but emotionally distant. Questions about people who lived so long ago that we cannot easily feel the weight of their lives. Slave ship DNA is recent. These are not people from 10,000 years ago. These are people from 300 years ago, 200 years ago. Some of them had living grandchildren within recorded history. The Clotilda's last survivor, a man named Cudjoe Lewis, who was among 110 Africans brought to Alabama in 1860, died in 1935 at the age of 94. A man who was born free in West Africa, kidnapped as an adult, and transported on the last American slave ship lived long enough to be interviewed by the writer Zora Neale Hurston, long enough for those interviews to become a book published in the 21st century. The DNA work being done on slave ship remains and burial sites is not filling in the distant past. It is filling in the recent past. It is answering questions that the descendants of enslaved people have been asking for their entire lives. It is, in the most literal scientific sense, restoring identity to people whose identities were systematically destroyed. Hannes Schroeder, reflecting on the St. Helena project, said it plainly: genomic information represents a new source of historical record. Through molecular analysis, it becomes possible to reconstruct and narrow down the range of possible source populations in Africa to give back, even partially, the knowledge of where someone came from. What does all of this mean? Taken together, it means that the slave trade, which erased so much, did not erase everything. It could not erase what was written into the hardest tissues of the human body. The DNA stayed, in teeth buried beneath Caribbean streets, in bones laid in South Atlantic soil, in sediments settled at the bottom of a burnt and sunken ship in an Alabama river, and in the living bodies of tens of millions of people across the Americas, whose ancestry maps trace back to specific regions, specific ethnic communities, and specific coastlines in West and Central Africa. It means the records always existed. They were just written in a language that took science centuries to learn to read. And it means that for every person alive today who carries African ancestry from the Americas, every descendant of the slave trade, every person who has ever stood in front of a wall in their family history and found nothing on the other side. Science is slowly, painstakingly, and with extraordinary care, building a bridge. Not a complete bridge, not yet. The databases of African genetic profiles still need to grow. The ancient DNA extraction methods still need to improve. The legal and political frameworks around working with remains of enslaved people still need to evolve, with descendant communities at the center of every decision. But the direction is clear. What the slave trade tried to take forever, DNA science is beginning to give back. The story of what's been found in these bones is far from finished. If this video moved you, share it. These stories deserve to be heard. Subscribe, and we will keep following the science wherever it leads.